Yeah, good morning everyone. Good morning. Okay, good morning everyone. Good morning Abdul. Good morning Apurva. Good morning everyone. Good morning. Okay, so friends, now in this particular class, it will take at least 3 to 4 days because we have to solve many questions. At least we have to solve 100 to 125 questions. We have to solve, including the part B also, as well as including the part C also. Okay, good morning everyone. So friends, I want every one of you has to treat this as an important paper. So what I want you to do is, I want you to spend at least 30 seconds when it comes to the part B question. When we are solving the part C question in the next class, we will spend 60 seconds on a given question. Okay. So without having any kind of a hurry, without making a wrong, without choosing any wrong option, just think it and give me the correct answer. Okay. Okay, your time starts now. Please go ahead. I will explain all the options briefly and elaboratively. Okay. Question ID 21. Good morning everyone. Please continue. No, it's not passer Sumaya, it's good. Okay. Now friends, you have to look into this. They have asked a question about the uncompetitive inhibition. So friends, if you know the characteristic features of the uncompetitive inhibition, undoubtedly you can answer the question very easily. Okay. Everyone, it's right or wrong, please give me the answer. By that I'll explain you. Think it correctly and give me the answer. Following statements are made about uncompetitive inhibition of an enzyme. Time up, everyone give me the answer. Following statements are made about uncompetitive inhibition of an enzyme. Okay, so friends, anyone can give me the answer fast. Please don't take too much of time. You're taking more than 30 seconds. Make sure that you will give me the answer within 30 seconds because it is the part B question. So you no need to spend greater amount of time on the question. Okay, of course you have to spend the time but not too much. No one is not thinking properly. Good morning Sushma. Okay, all the students pay utmost attention, my friends. Uncompetitive inhibition. Saima, remember, Abdul, remember what we studied in the uncompetitive inhibition. In the uncompetitive, every student listen. You can easily eliminate the option. See, I want every student has to remember one statement. In case of a uncompetitive inhibition, the inhibitor never ever, please listen, never ever bind to the free enzyme. First statement every student has to remember. The uncompetitive inhibitor never associated with the free enzyme. So this is the free enzyme. To the free enzyme, inhibitor never associate. Now the point is, my friends, the uncompetitive inhibitor associated with what? Uncompetitive inhibitor only associated with the enzyme and substrate complex. This is what, my friends, this is the enzyme substrate complex. Only to this uncompetitive inhibitor will come and attaches. Then every student answer my question, how come the option A will be the right answer? which is absolutely wrong. No one is not thinking properly. It is a direct question which has been asked from the characteristic features of the uncompetitive inhibition. At least remember now, an uncompetitive inhibitor never ever attaches to the free enzyme. An uncompetitive inhibitor only attaches to the enzyme substrate complex only. That's the point you have to remember. But what was given in the option A, we read the properties. Uncompetitive inhibitor binds to both free enzyme as well as the enzyme substrate complex, which is the wrong statement. You can easily eliminate option B, option C, option D, because the option A is a wrong option. Friends, slowly, that's what I'm saying to all of you. Reading is different, revision is different, practicing is different, answering question is a skill. So slowly analyze. Of course, friends, when we add the uncompetitive inhibitor, there will be a change in the Vmax and there will be a change in the Km. At least remember now. There will be a change in the Vmax, there will be a change in the Km. So what is the point you have to remember? Good morning, Shafia, continue. So addition of an uncompetitive inhibitors lower the Vmax of a reaction. Of course, true statement. At the same time, the Km also get lowered. Hence, my friends, option B is correct, option C is correct. In a given option D, they said that apparent Km of the enzyme remains unchanged. This is wrong statement because it was clearly said that Km is going to be changed. There is no doubt about it. Hence, my friends, see, this question is depending on the luck also. What do you mean by the luck here? At least if you can predict whether the statement A is correct or wrong, undoubtedly you can predict you can come to the answer, that is the B and C. See, since you are unable to predict this, it will become difficult for you. And the question is not at all indirect. The question is directly asked from the characteristic feature. And I want every student has to look into the explanations without fail after the class. Uncompetitive inhibitor bind only to the enzyme substrate complex, not to the free enzyme. Hence, the statement A is absolutely false. Both Km get altered, friends. Vmax also get altered. So don't get confused. Hence only B and C are correct. Hence the correct answer will be the A. Don't worry. No problem at all. Okay. Remember see, suppose example, please listen. If you solve 50 questions, if you answer 30 questions correctly, suppose example, if you answer 35 questions correctly, out of 30, if you answer 35, out of 30 are correct, which indicates that your preparation is 100%. There is no rule that you have to answer 50 questions. That is not the thing. If you answer 35, at least 30 should be correct, and at least 28 should be correct. Then I can say that your preparation is 100%. Okay, don't worry. Okay, next one, question number 2. See, now only we will understand how many questions asked from every chapter wise. By that we will get a very good idea. They have asked for the amino acid lysine. Twenty seconds more. Requesting all of you to do little faster because I want to solve more number of questions. First, what you have to do, you have to understand what is lysine and how many forms it can exist and at which form it will be exhibited in the neutral or where it is exhibiting in the Zwitter ionic form. Others, okay, very good. So friends, we all people know, right, in order to calculate the pi of lysine, first of all every student has to remember that friends, lysine containing the R group, which is the ionizable group. Yes or no, friends? Lysine having what? Lysine having an extra amine group, Correct na? Lysine is a what kind of amino acid? Basic amino acid. We all people know that it is having the extra amino group. If you calculate at which point it can exhibit in the deuterionic form, by considering the value of pK2 as well as the pKr. I want you to remember it compulsory. So what will be the pK2 value? 9.06 as well as the 10.54. You know how to calculate it easily. By 2, how much we will get? We will get the 9.8. Hence the correct answer will be D. Simple, over, easy question, direct question, no need to confuse at all. Now Sushma, please look into that, dear. The answer will be the D. Okay, hope everyone understand. Okay, now friends, don't get confused. I want every student has to give the correct answer for this particular question. I want you to apply the Michaels-Menten equation. Saima, do it correctly. Priya, Archana, everyone has to do it correctly. Sumaya, make sure that you will do the correct question, correct answer for this particular question. Just apply the Michaels-Mentens. You don't need to do anything at all. Everything was given. You have to just multiply that. That's all. You don't need to do anything. It's a direct question. Everything they have given. Easy question, directly you can answer. Okay, so friends, what were given in the question? The enzyme alkaline phosphatase was tested for its catalytic activity using the substrate papa nitrophenyl phosphate. The Km obtained, what is Km? Km is the Michaels-Menten constant. Vmax also they have given, final velocity. And which one of the following option represents the initial velocity of the reaction at a substrate concentration at 10 millimolar? That's the given question. So friends, what was given? They have given the Km. What will be the Km? 10 millimolar. Correct now friends? Anyone has? Okay, good, Vinita. Okay, and what else has given, friends? Every student has to understand. They have also given the Vmax. How much Vmax they have given? 100 micromolar per minute. Correct now friends? And what they have given the substrate S, which is the 10 mm or 10 into 10 power minus 3 micromolar. What you, instead of doing all this, just multiply whatever you have given in the Michaels-Menten equation. Just multiply it. So what is the Michaels-Menten equation? Just remember, V naught equal to V max S Km plus S. This is the Michaels-Menten equation. I want you to multiply that, nothing else. So listen. So V naught equal to, what will be the V max? V max 100 into 10 into 10 power 3 by, so what we can say here, 10 into Km into 10 power 3. Very easy question. I want you to do the calculations. Anyway, you will be having virtual calculator. You can easily answer the question. By 2 into 10 power 4. Obviously, you will get the answer of 50 micromolar per minute. Very easy question. Okay, what you have to do is the Michaels-Menten equation, because friends, they have given Vmax. What is the Vmax? Final velocity. What is S? Substrate concentration. What is Km? Michaels-Menten constant. And what is the question they have asked? They asked us the question initial velocity. They have given the maximum velocity. They asked us to calculate the initial velocity. So just apply the Michael-Menten. Obviously, you will get the answer, which is the A. Very, 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 very easy question. Direct question. You have to just apply the Michael-Menten. Okay. So the answer was given. If anyone having any doubts, look into it and ask me if you have any kind of a doubts. Next question. Archana, follow it. Even though you are unable to answer, please try it every single question. Okay. Next, question number 24. I think in the yesterday class also we studied. How many hydrogen bonds involving in the backbone of C bond NH can be observed in an alpha helix consisting of 15 amino acid residues? So I want you to remember only this: I to I plus 4, ith amino acid to the I plus 4 amino acid. Based on this, you can calculate. Yes, yes, yes. Focus, focus. Answer is B. Okay, anyone else? Friends, the answer is very easy. There are total 11 number of amino acid. Correct na? Friends, you all people can quickly look into this. The first hydrogen bond will be I plus 4. Then friends, you can easily remember I plus 1, I plus 5. Very fast I am writing. I plus 2, I plus 6. Then friends, I plus 3 to I plus 7. I plus 4 to I plus 8. Then friends, I plus 5 to I plus 9. And I plus 6 to I plus 10. Then friends, I plus 7 to I plus 11, as well as I plus 8 to I plus 12. Then I plus 9 to I plus 13. Very easy to calculate. I plus 10 to I plus of 14. So friends, total 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 hydrogen bonds are involved. We all people very clearly know that. I want every student has to listen my statements very carefully. The first 4 NH groups, and last 4 CO groups are not involved in the hydrogen bonding. This is the statement every student has to remember for the exam. The first 4 NH group and the first 4 CO groups, last 4 CO groups are not involved in the hydrogen bond. What is the reason? Because they are not, the amino acids of this amine group and carboxyl group, they are not completely involved because they are having partial positive charge and partial negative charge. So you can easily remember 15 minus 4, which is the 11. Easily you can remember that particular answer. Very good, very good. Okay, so friends, by the biochemistry is over. So how many questions asked in the biochemistry? 21, 22, 23, 24. 4 questions were asked in this, 4 to 8 marks. Now, is there any necessity that we have to answer all the 4 questions in order to crack the exam? No. At least out of 4 questions, it makes sure that you will give 2 questions correctly. If you are behind of answering the 4 questions correctly, you are doing the biggest blunder mistake. That is not the idealizer quality. If you want to get a good score, it's not required because we are not dependent on all the chapters. We have dependent on only few 10 chapters or 11 chapters. So we can give the try in every single chapter. So my friends, out of 4 questions, make sure that you will answer 2 questions very correctly, 100%. That is how you have to do it, friends. Okay. Question number 25 starts from cell biology. We already studied it because we have already studied in the cell cycle regulation. It was mentioned a particular table. In that table only this particular answer was there. We have already studied this. Okay, since it's the direct question, you people no need to remember anything. Okay, so which is this cyclin E? Okay, cyclin E is the one which is basically helpful in entry of the cell into the S phase. Obviously friends, CDK2 cyclin E, hence the correct answer will be the B. Very good, very good, fantastic. Okay, then the second question, question ID 26. We have discussed this particular question in the cell biology chapter. In the cell biology chapter, when we are discussing the plasma membrane, I have explained you this. I have explained you while I am explaining the plasma membrane. Slowly friends, slowly. You know, here there is no subject is lying here. There is no concept. You have to remember that. No problem friends. Always remember that. Anyway, you know the answer. We have discussed, it was there in the slide itself. Myristic acid, palmitic acid. During the process of a post-translational modifications, friends, they will be added to the sugars. Correct, na? Where exactly they will add it to the sugar? The alpha amino group of a glycine. Keep it in your mind. The alpha amino group of a glycine, it is the one basically associated with this particular myristic acid and palmitic acid. By which linkage, friends? Of course, by the covalent linkage. Obviously, the answer will be the glycine. Very straightforward question. Here, the question is not at all difficult, which is direct asked question, which you have to remember. That's all. Okay. This is also does this question is with respect to the concept oriented? Not at all, which is a very direct question. I have explained it to you many times. Please don't say that I did not explain. It was there in the questions itself. It was written that the movement of protein bigger than 50 kilodalton across the nuclear envelope requires. Very simple, very straightforward, very direct question, which we have studied it. By looking at this also, you can eliminate it. Friends, we have studied, right? Hello Sidhu. We have studied now, friends, Tim and Tom. These two are with respect to the mitochondria. So you can eliminate the option D, you can eliminate the option B. Sec61 do not have any kind of a role. We already discussed that there are two type of importins are there. Friends, please don't get confused. Even though they give the importin alpha and importin beta, keep it in your mind, friends, both alpha and beta both plays a key role in bringing the proteins into the nucleus via nuclear pore complex, which is more than 50 kilodalton. Suppose example, instead of importin beta, if they give an importin alpha also, that is also absolutely right. Don't worry about that. Okay, perfect. Next, iron sulfur cluster or the Cre prosthetic groups that carry electrons in all of the below except. Very good question it is, and direct question. It is also direct question. It is not at all belonging to the any concept. You have to remember. You don't need to apply it. You don't need to analyze the question. Direct question. Yes or no? They are asking except, you have to tell me except. Which do not have iron sulfur cluster. That's the question you have to tell me. You read in the biochemistry also, even you read in the cell biology also, even you seen this. Okay, slowly friends, slowly friends. Slowly, don't be confused. See, most of the students are not understanding the given question. Slowly, friends, listen to me carefully. What is the question asked? Sumaya, please look into the question. What is they asked? Saima, look into the question. Priya, look into the question. Iron sulfur cluster, FES. Shafia, what is the answer you have given for question number 28? Iron sulfur clusters are the key prosthetic groups that carry electrons in all of the below except. What is the question asked is, there are 4 electron acceptor complexes are given. What are all these 4? They are electron acceptor complexes. What is the question asked is, among these 4, which of the complex do not uses iron sulfur cluster as their prosthetic groups? That is the question they asked. Which of the complex do not use the iron sulfur cluster? That is the question they have asked. The answer will be C. Correct, Shafia. Only Shafia has given the right answer. Friends, the cytochrome C oxidase will uses what? The cytochrome C oxidases always uses heme and copper ion. It will uses heme and copper ion as their cofactors or are their prosthetic groups. Whereas NADH and succinate and cytochrome C reductase, these 3 obviously uses iron sulfur as their cofactors. Hence, my friends, the correct answer will be C. Please don't get confused. Correct answer is C. Option A, option B, option D is wrong. These 3 will use as iron sulfur cluster. Iron sulfur cluster only cannot be used by the cytochrome C oxidase. Instead that it will use the heme and the copper. Simple. Answer is C. Don't be hurry. Don't be hurry. Okay. So friends, we have understand that question number 21, 22, 23, 24. These 4 are from the biochemistry. So we got 4 part B questions. And look into the 25, 26, 27, 28. These 2 are from unit 2. What is the point you have to understand by analyzing these 4 questions? You have to understand one point, that the 4 questions are yes or no. Not at all analytical, not at all experimental, not at all graphical. Very straightforward questions which are yes or no. For this particular kind of questions, what you will be requiring to answer these questions, you have to remember the concepts for the longer time. And the surprising factor is that none of the question is an indirect question. Every question is a straightforward question. So most of the students will answer the questions which are concept oriented. Most of the students never answer the questions which are straightforward because they never read that. So that's why remembering concept is very important. Don't get confused. Okay, from here it will start what? Molecular biology. Slowly, my friends, please analyze the question. I want every student has to answer this question very correctly. Take your time, analyze the question properly. Slowly, friends, slowly. It's an easy question. Please give me the answer. Shafia, what will be the answer for this? Sushma said B. Okay. Anyone else? You know friends, for this question, even if you read the Pathfinder also, you can answer it very clearly. Even in the Pathfinder also, the answer was very clearly given. Okay. All the students, please look into the answers carefully. First, look into the question. Everyone pay attention. Ask me if anyone did not get it. In bacteria, many of the tRNA genes do not contain the CCA sequence found at the 3' end of the tRNA. In this context, which one of the following statements represents the correct explanation? Friends, tell me. This is the tRNA. Correct na? We all people studied very clearly that the 3' end of the tRNA is the one where the amino acid will come and attaches. That is absolutely true. Please listen, follow me correctly. Now we know very well wherever the amino acid is attaching at the 3' end of the tRNA, friends, there is a specialized sequence is present. What do you call it, that particular sequence, friends? We will be calling that particular sequence as the CCA sequence. Now what every student has to understand is, all the students listen, this is the gene, this is called as tRNA coding gene. Please listen friends, this is the gene which is giving rise to the tRNA. There is no doubt. Once again listen to me. This is the gene which is giving rise to the tRNA. Now my question is very clear. When this particular gene is giving rise to the tRNA, do you think this gene can code for the CCA sequence? No, not at all. Very important point it is. Once again, please understand what I am talking, friends. That tRNA coding gene never ever code for this particular CCA sequence. This is what I want you people to remember. There is a specialized polymerase enzyme. We can call it, what is that polymerase called as? CCA RNA polymerase. There is a specialized enzyme called as CCA RNA polymerase enzyme will come and add this particular CCA sequences to the tRNA. So this will be the answer. Please remember friends, you have to remember this enzyme is a very specific enzyme. What do you mean by the word specific enzyme? This CCA RNA polymerase enzyme do not have any other function except adding the CCA to the tRNA. That's all. It do not have any function. So now you only look into the given options. In these, tRNA amino acylation occurs at the 3' end of the tRNA irrespective of the presence of the CCA sequence. No. What do you mean by the option A? Option A is also very confusing option. They are saying that without addition of the CCA sequence, protein synthesis will occur. That is absolutely wrong. If there is a no CCA sequence, you have to listen. If there is a no CCA sequence, first of all, tRNA is unable to associate it with the amino acid. Hence, this option is a wrong option. Option B, CCA sequence is added to this tRNA transcripts in a DNA template independent manner. Hence, this will be the correct answer. What will be the enzyme? CCA RNA polymerase enzyme. Do not get confused with that. And look into the option 3. These tRNA exploit the process of transplicing. You tell me, what is transplicing? Transplicing is a splicing which is occurring between the two different mRNAs. We are talking about the tRNA. Then why the trans-splicing has come? This option itself wrong option. And option D, the absence of CCA sequence occurred only in the last common ancestor. That is absolutely wrong. There is a no role of the CCA to the process of evolution. There is a no role, or there is a no relation. Option D is also wrong option. Hence the correct answer will be, friends, undoubtedly option B. Very simple question. Don't worry, explanation were given. Don't worry. Good morning Sanjay. Okay, I hope you understand correctly, friends. Please don't get confused or ask me if you have any kind of a doubt. Even if it's a small doubt, please ask me. Yes. Question number 30. Question number 30. It's easy. It's very easy. In the 2 days back we have seen the amino acids. In that also I have written this particular D form of amino acid. It was directly come from there. It's a good question. Take your time. Anyone can give me the answer for question number 30? Analyze very correctly. Option B. Slowly, friends. Slowly. Don't be hurry, please. Okay. What will be the given question? The question says that some cell possesses peptides which contain D form of amino acid. How do they arise? We have very clearly discussed that, friends. If you look into the prokaryotic organism, it majorly contains D type of amino acid. But when you look into the eukaryotic organism, good morning Deepika. Deepika, without fail watch the recorded, do not miss it, okay? Without fail watch it. In case of a eukaryotic organism, we studied, friends, mostly L amino acids will be found. Okay. Now the question is very clear. They did not ask anything about the L amino acid. Then neglect this. The question asked is about D amino acids. Friends, I told you right. D amino acids which are synthesized. Please remember, during the post-translational modification only they will be involved. During the post-translational modification only they will be involved. Which means that in order to synthesize you have to listen to me carefully. In order to synthesize the proteins, friends listen, in order to synthesize the proteins from the D amino acids, there will be no requirement of a ribosome. Once again listen. In order to synthesize the proteins from the D amino acid, there will be no requirement of a ribosome. All the proteins which are developed or which are synthesized from the D amino acid, they are always synthesized in a ribosome independent manner. Do not get confused with this particular point. The peptides with D amino acids are produced in a ribosome independent manner. Never ever confuse. Hence option C is the correct. These peptides are produced by ribosomes by incorporating D amino acids at specific position. Wrong statement. And most of the people has given the answer as B. Ribosomes make peptide with L amino acids only. However, some of the amino acids in the peptides are replaced by the D amino acid. No. You have to remember that D amino acids never ever replace L amino acids. That's not possible even. See, remember, one type of amino acid cannot be replaced with another type of amino acid. You only tell me, do you think, friends, basic amino acids can be replaced by the acidic amino acid? Is that possible? In other words, acidic amino acids can be replaced with the basic amino acids. Is that possible? It is not at all possible. What is only possible? What is only possible is that one organism specifically contains L and one organism contains specifically D. If we want to synthesize a protein, if we want to synthesize a peptide with L amino acids, we will be requiring the ribosome. If we want to synthesize the D amino acids, we do not require the ribosome. Then how this particular they will be involved? This D type of amino acids produced in the ribosome independent manner during the post-translational modification. That answer will be the C. Don't worry, don't get confused. Just think and answer the questions. Don't worry. Next question. Question ID 31. They are asking except. Archana, you have to answer this question correctly. Question ID 31. We read that. My friends, please look into the option. They have asked except. Don't forget the except. Take your time. If you remember one statement, you could answer the question easily. Saima, what will be the answer? Shafia? Shushma, what will be the answer? Abdul? Friends, we discussed it very clearly. It's an easy question also. Sia. They are asking except. Abdul, they are asking except. They are asking except. Saima, what is they have asked except? Okay, all the students listen. No, slowly friends, slowly. Don't be hurry, please. Termination, listen. Termination is the one which is depending on two types. One we discussed that intrinsic termination. One we discussed that, listen friends, extrinsic termination. Correct na? Intrinsic termination, look into this Saima Abdul, row independent termination, and extrinsic means what? Row dependent termination. Correct na? Now, in case of a row independent, what will happen? Hairpin loop will occur. Correct na? Here also hairpin loop will occur. There is no doubt about it, but there is a protein required is the row. Now question is NUS A. What is NUS A? Every student has to remember. NUS A in one line, remember, which is the anti-termination protein, which never allow the termination to occur. What protein it is? Anti-termination protein. In other words, we can say that termination, friends please follow me, termination passing protein. Termination passing protein or the anti-termination protein, that is called as NUS A. If NUS A is present, termination never occur. But what is the question they asked? Which is true for the termination? Then why we consider the NUS A? And please remember, NUS A is not present in the body. NUS A we will only use in the laboratory, in vitro, within the laboratory. This is the one which will stop it. Now look into the option A. Some terminator sequences are row protein for termination, right? Some required row. Inverted repeats of T-rich non-template strand define the intrinsic terminators. Absolutely true statement, even option B also. See friends, we discussed A and T will form the hairpin loop. They will only helpful in the termination. See friends, first statement explaining about extrinsic termination. Simple point. Second statement explaining about the intrinsic termination. Very simple point. The third statement saying that row dependent terminators may possess inverted repeated elements. Yes or no friends? I have clearly told you, right? Friends, even though row protein is required, row protein is present, hairpin is important. Because row protein will travel near the hairpin, it will provide the enough strength to the hairpin to pull it. So row dependent terminators also contain hairpin. That is the inverted repeat element. Don't confuse. Now you look into the option D. NUS A protein is necessary for termination. No. If the NACA protein is present, there will be a no termination. It will start the anti-termination. It never allow the termination to occur. Hence, the wrong option will be option D. I hope every one of you understand without confusion. Archana, NACA is the anti-termination protein or the termination passing protein which is exclusively used in the in vitro or within the laboratory. Got that? Okay, perfect. I hope everyone understand the question without confusion. Okay, we all people know this very well. Which one of the following proteins is essential for the both initiation of DNA replication as well as the continued advance of the replication fork? It was also written in the slide. It is a very easy question now. Which one of the following is essential for the both initiation of DNA replication? Okay, we discussed that, friends. CDC45. See, friends, sometimes instead of giving the CDC45, they can use the word GINS, GINS. Even this also are right. Okay, you don't need to remember the full form. Instead of giving the CDC45, if they given GIN, that is also absolutely true. These two will associate with the MCM. They will activate the MCM. Then later MCM will get phosphorylated. Then the process of initiation of replication will occur. Hence the correct answer will be C. Wonderful, wonderful, wonderful. Okay, okay. Next. And friends, most of the students will neglect the eukaryotic gene regulation. Most of the people will neglect the eukaryotic gene regulation. See the question asked from the eukaryotic gene regulation. They are asking combination. Very simple, friends, which are required for the chromatin condensation. What is chromatin condensation? Which is the heterochromatin form. In the heterochromatin form, which proteins are required? That's all. Very simple question. It's a direct question, friends. You don't need to think about it. We all people know, right? When there is a heterochromatin, the entire process can be blocked. There will be a no gene expression. There will be a no transcription will be taking place. The answer will be the B. SIR protein complex and heterochromatin protein 1 complex. These two are the major proteins which make sure that the gene expression never takes place. In order to block the gene expression, what they are doing, friends, they will make the DNA to convert it into a condensed form, chromatin form, which is the heterochromatin form. The answer will be B. Here nothing to read, everything you have to remember, which is a direct question. Okay, so friends, you only listen. How many questions were asked in the unit 3? 29, 30, 31, 32, 33. 5 part B questions were asked. So 5 into 2, 10 marks. How important it is? How important it is, right? Hence, you should not neglect. Is there any necessity that we have to answer every 5 questions? No. What is our target? 2 questions. So far we have done 3 chapters. Unit 1, Unit 2, Unit 3. From Unit 1, Unit 2, Unit 3, there are total 15 questions were appeared. Is there any necessity to, 14 questions were appeared. Is there any necessity to answer 14 questions? Not at all. Our goal is to only answer 6 questions. In this way only you do. Don't try to answer every question which you are not sure. Okay, good. Now here onwards, unit 4. Unit 4, the question starting with which one? Host pathogen interactions. Very direct question. The question asked is which is the causative agent for the filariasis. Very direct question. We all people know that filariasis can be caused by what, friends? Parasitic roundworms. Correct na? Parasitic roundworms are the one which is basically causing this particular infections. What are the two organisms that are involved here? We all people know that Wuchereria bancrofti. What, friends? Wuchereria bancrofti, as well as, friends, Brugia malayi. Brugia malayi. These two are the organisms basically cause this particular filariasis. Anyway, they did not given the Wuchereria bancrofti, they have given only Brugia malayi. Very direct, straightforward question. To be honest, you read this filariasis even in the NCERT also during your plus 12th. There is a chapter called as Human Health and Diseases. There also you have studied this. Very direct question. Okay. We all people know this. Yes, question number 35. Very easy question you can answer. Many number of times we discussed it. What will be the answer for question number 35? Many times we discussed. Okay, so friends, let's take an example of RTK receptor. RTK receptors can be used by what? Many growth factors. An example, epidermal growth factor, insulin growth factor, nerve growth, many times. Nerve growth factor, platelet derived growth factor. We have studied all these. So friends, you all people know, right? How does the RTK structure look like? RTK contain extracellular domain, transmembrane domain, and cytosolic domain. We all people know, friends, the cytosolic domain having the kinase activity. So epidermal growth factor uses that, platelet derived growth factor uses that, insulin growth factor uses that. Now I want every student has to remember what's the function. What is the ACL glycoprotein. Please remember, the moment you heard the name of this, every student has to remember that this receptor is only with respect to the process of endocytosis. Next time when they ask any kind of a question with respect to this, always remember that acetylglycoprotein is exclusively with respect to the endocytosis. Always remember that particular point without fail, right? Hence, the correct answer will be the D. Very good, very good, very good. Okay, next. Question number 36. The tumors are generally classified as. The tumors are generally classified by. Fast, friends. Anyway, I will only give the answer very fast. Yes. Correct, Sushma, correct. So we know, right, friends, the tumors are basically classified from which origin they are derived from. The tissue or cell origin. Correct na, friends? We will say that there are many cancers are there. Breast cancers are there. Correct na? We can call lymphomas. We can call melanomas. We can call sarcoma. Sarcoma, melanoma, lymphoma, breast cancers, ovarian cancers, brain tumors. All these names were given based on what? It's not based on the metastasis, because every cancer will show the metastasis. It is not depending on the person discovered, not depending on the virus caused. It is basically from the tissue or the cell which is originated. Okay. I want you people to think this and give me the answer. Don't be hurry, don't be hurry, don't be hurry. You know, this is the recent technology. Don't worry, I will give the answer for this. Focus. A breakthrough in a cancer therapy is expected where T cells are taken from a patient or modified in the laboratory to attack cancer cells before the reinfusion in the patient. These cancer cells called as what? And first of all, this technique we can call CAR T cell therapy. No. We can call this technique as the CAR T cell therapy. What is CAR? Chimeric antigen receptor. No, friends, listen. What we do, you know, from a patient, we will take the T cell. Please listen. From a patient, we will take the T cell. After taking T cell from a patient, what we will do, friends? We will modify the T cell. We will modify the T cell. We will modify the T cell in such a way that which recognize the cancer cell and destroy that particular cancer cell. See, we will take out the T cells from our body. We will modify that in such a way that it specifically recognizes that. That is only called chimeric antigen receptor T cell. Answer will be the B. Very good, very good. Okay. So friends, how many questions were asked? So 34, 35, 36 and 37. So 4 questions were asked in the unit 4. 4 questions were asked in the unit 4. So 4 into 2, 8 marks. What will be your target? Your target should be 2. Simple. So friends, if you look into the unit 1, unit 2, unit 3, unit 4, unit 5, including these 5 units only, you will get 22 part B questions. Minimum. I am using the word minimum. Out of this 22 part B question, every student has to remember your target should be minimum 12 questions. I am using a word, your target should be minimum 12 questions. Okay, perfect. Okay, now question number ID 38 from the unit 5. I don't know whether you believe it or not. This entire question will be directly from the slides of our PPT only. Direct question. If anyone follow my lectures of developmental biology, you could feel that the same lines we have studied. Same lines. Saima, what will be the answer? Priya, give me the answer. We have studied many times this. Saima, what will be the answer? Deepika? Saima, how it will be C, dear? How it will be C? How direct a question it is? What is the beta catenin? I told you many times. Correct or not? Beta catenin. Every student, please remember how it is C. I am asking. Beta catenin is an important factor. What is the function of a beta catenin, Saima? Beta catenin is an important factor which is responsible for the formation of a Newcomb center in the dorsal most vegetal cells. Dorsal most vegetal cells formation, we require beta catenin. Tell me, Simon, why we require the beta catenin for the organizer? It is not required. Very good, Sushma. Correct. Now, if you look into the option B, organizer induces the Newcob center. That is wrong statement. Newcob center induces the organizer. Opposite. Newcob center induces the organizer. In the absence of BMP inhibitor, ectodermal cells form neural tube BMP. No, that is wrong. Look into this. Look into the diagram which we have discussed in the classroom. That first diagram, the same line we have discussed. Gastrulation begin with the invagination of a bottle cells. We discussed, right? The bottle cells will look like this, followed by the involution of the mesodermal cell. Then finally epidermy of the epibole of the ectoderm. I have explained 7-8 times this particular thing. Correct answer will be the A. Friends, they have given the direct statements of the gastrulation. In case of a amphibian, direct statements they have given. Newcob center induces organizer. This is wrong. Organizer is, see, it was written very clearly. I want every student look into the explanation later without fail. The major candidate for the factor that forms the Newcob center in the dorsal most vesical cell is the beta catenin, which is a wrong statement. They said that the organizer is formed by the beta catenin. That is wrong. Organizer never formed. The Newcob center formed by the beta catenin. Easy, friends. The question A will be the right. No problem, no problem, no problem. I am requesting all of you without fail, go through the PPT and look into the explanation without fail. Okay. So once again in the tomorrow 11 o'clock we will try to finish the entire part B questions. We will finish the part B, then we will move on to the part C. Okay. So friends, that's all for now. I'll upload the PPT. Please look into the PPT in the app. And 3 p.m. we will meet. We will start the enzymes and its kinetics. Okay. Okay, friends, without fail, please revising. Okay. I'll see you at sharp 3 p.m. in the enzymes class. Okay. Thank you all. Thank you so much. Hope the discussion is little useful to all of you. Bye-bye. Thank you all.